Real World STEM wants to know, did you take the bloodstain challenges? Ellie, Kenzie, Jack, and Clay did, and here's part 1.
It starts like the paint gets like thinner or thicker as it goes. Well, it gets thinner, like width.
It's a bit wider, and it's still moving, but it's like more. I can't think of the word. Yeah, like it's not as run out yet. Like it's still, there's more paint in a smaller area.
A lot more of like a long strand, because the board was like at less of an angle. It got more time to like go all the way down. And like there's some off the side of it, like where it might have hit before and kept going. It's much thinner than the other drop.
There was a lot more scratch, like a little more drop. So, but how can you tell if the blood came down straight or sideways, or if is there any specific signs, or like did it start getting thick at one side and then thin out? The splatter start to thin out the other way? Or?
Clay develops a hypothesis based on the experiment.
If she's like there, she sees blood like that.
Yeah.
Compared to blood like this.
Yeah.
She can kind of like get an idea of like how much blood was spilled and like where it might have been coming from, because this, it could have been over here and like splattered, but here, like definitely from on top over here.
From the actual stain, we can tell certain things. Where that sample actually came from. Was it from the right side, the left side, up, down? We can also tell other things like from the angles, where the point of convergence is, where that blood originated from. If it was from, let's say somebody was being beat in the head, and then you have the impact spatter on different surfaces at different angles, you can use those angles and calculate back to where that head was. And by doing that, you can either help corroborate or refute a statement made by maybe a witness or the victim or the suspect. I'm what's called a senior criminalist in the Boston Police Crime Lab. We are called to crime scenes if we're needed. We do collect evidence. We do bloodstain pattern interpretation, chemical enhancements. We'll collect evidence, bring it back to the lab. Then we get assigned cases and we testify in court to our results and to our opinion. If you have a 90-degree surface, so a flat surface, the drop just hits the surface and you get a very circular stain, depending on what type of surface it is, whether it be glass or corrugated cardboard or anything. But if you have an angled surface, the steeper the angle gets, so basically the smaller the angle gets, so anywhere from 90 to 0, the stains get more elongated, so more oval, and they also have more of this satellite spatter coming off the bottom of it. So you can tell, one, that this was dropped from up above because you have the pooling down here, as well as the satellite spatter coming off of here, off of the parent stain. But you can also tell, because of the elongation, you can calculate back the actual angle that this paper was at when the blood hit it. It's a geometrical angle calculation that you do. So they're very easy. You can do them on a calculator, but you have to understand that the width and the length of the stain are actually part of that SOHCAHTOA triangle that you learned about in math class.
And that's trigonometry, right?
Yes.
After the angle experiment, we dropped fake blood on different surfaces. Here's what happened.
Since it doesn't have much to hold it up and it's very flimsy and it's somewhat slippery, the paint can spread on it, but compared to this, it's cushiony and the cloth absorbs the paint, so it doesn't run as much.
Alright, both of my objects are very hard. Not like a trash bag or the oven mitt. They were about as, same kind of splatter as on the wood. except for the seashell, which had like little ridges, so it was able to like pool up in the ridges.
Both of my objects were very flat. I don't really know how that happened. It's like in a perfect circle. And then this one, I expected it to splatter because it's glass.
Both of my materials were very absorbent. This one more splattered, so the paint didn't really, it doesn't move a lot. They just kind of absorbed it, but like this one's not as absorbent. This one's just cloth, so it soaks right through, but this takes time to absorb.
As the blood hits a certain surface, it displaces itself. If it hits a smooth surface, it will just basically displace itself out to a circle. If it hits a more rough surface, then that rough surface takes the blood and little tiny droplets from that parent stain, the main stain, actually get displaced out as satellite spatter. The edges of the stain aren't as circular because of the rough surface. If you have something that absorbs the sample, then the stain is not only going to be smaller, but it's also going to have a little bit of a rough edge, but not as many spines or the elongated spatter. It's going to have more of the droplets that go out in the satellite spatter.
By the blood splatter, could you tell like how high like the blood came from? Would the splatter be bigger or smaller?
This is from 4 inches, and this is from 36 inches. So it's on the same surface, but you see that this stain is smaller than this stain. If it's very close to the surface that it drops onto, you get a much smaller stain than farther and farther away. However, there is a point where, you know, basically the volume of blood can only create a certain size stain on a surface that it's hitting.
When Amy's not at a crime scene or testifying in court, she's here.
This is what we call the serology lab. This is where most of the evidence is examined. Weapons, clothing, headboards, guns, anything that we have to examine to see if it pertains to the crime. If we have the suspect's clothing or anything like that, we want to look at that first and then process it for reddish-brown stains, what we call reddish-brown stains. And then we test it to see if it is actually blood. If it's blood and we know that the suspect wasn't bleeding, then we send it into our DNA unit because it could be the victim's blood. We try to see if there's any linkages between the victim and the suspect and that physical evidence. We try to reconstruct the crime scene potentially, if we can, with bloodstain pattern, trajectory analysis, anything that can help tell the investigator what actually happened. And the third thing that we try and do with the physical evidence is try to give the investigator leads on potential suspects. So if there's a DNA sample that they don't have a suspect for and they get a profile, then it would potentially be linked to somebody that was already in a database, and we could give the investigator their name. Or if it's like a paint sample, a paint chip from a hit and run, we could potentially give the investigator information about what make and model the car is that they're looking for in a hit and run accident. Well, I'm part of the trace evidence section, and it's all the small stuff that's transferred on the commission of a crime. And it's usually more the chemistry side of the house than the biology side of the house. So we focus more on using microscopes and instrumentation than biology. So we have put samples on here that we want to create a mixture and not just have it be, you know, this component of that mixture is in this part of the tube and this part is on the other side. You want it to be mixed so that you can then centrifuge it to get that biological sample down at the bottom of the tube and then put it onto a slide to examine it on a microscope. We look at hairs and fibers and paint chips underneath microscopes, skin cells, sperm cells. We use flashlights a lot. And they're not the blue lights but that you see on TV, but the flashlights. They're just plain white light to be able to see contrast between what our sample is versus the surface that it's on. So this is a type of microscope that we might use to look for skin cells as well as sperm cells, and it is what's called a compound microscope. This is a 40x, so it's about 400 times magnification, and you can actually see the cells. They stain them before they look for them with what is called the Christmas tree stain. The nucleus of the skin cells are a reddish color. The outer area is green, and then the spermatozoa heads are stained red. So you can pick them out and then potentially send it on for DNA for a possible hit against an offender. When we're in the lab, if I have an item of evidence out or something on the microscope that I want to take a picture of to show a jury or the detective or the district attorney that stuff, then I'll take a picture of it in the lab. These are like second skin for me. We use these all the time. We want to protect ourselves against contamination from the evidence, as well as protect the evidence against contamination from our skin cells and everything. So it's just personal protective gear. Most of our DNA cases are breaking and entering, where somebody has left blood behind. But we get about 1,000 cases a year total. About 60 to 70 of them are homicides. About 200 are sexual assaults. And then the rest are anything from breaking and entering, or larceny, or assault and batteries, or hit and runs. I went to college for biochemistry, and then I got my master's in forensic science. So I had a lot of chemistry, biology, physics classes, math classes. There's several classes that I've taken afterwards during the course of my 12 years being here. I think it's about 3 or 4 bloodstain pattern classes where we do all of these experiments to see what we would actually potentially see in a crime scene and why, why those patterns were created, and from that, what is our opinion about what happened during the commission of that crime. So, doing these experiments helps us determine the reconstruction of a crime scene, potentially. I love the fact that every day I come to work and I don't know what I'm going to be doing that day. I could be called to court, or I could be called to a crime scene. I got into this field because I love science. I love everything about it, chemistry especially. With forensic science, it's a little different. You can come to a conclusion or to an answer a lot faster than with research. And I really liked that aspect of putting science with law and understanding that we can help out in a criminal case. That's what bloodstain pattern analysis does for us. It reconstructs the crime scene and gives information to the investigator or to the judge and jury when you go to trial about what potentially happened during the crime. It feels good to be able to tell the detectives and the investigators, this is what I think happened, and potentially this is what happened during the crime, and for them to go, oh yeah, that's probably what happened, and for them to be able to go to court with that, or for us to be able to testify in court with that, it's a good feeling.